We introduced LineageD, an interactive visual system to help biologists with cell lineage assignments for plant embryos. This process is needed because biologists take a single confocal microscopy image at some point in the embryo's development and then use the segmented data to deduce how the embryo developed up to this point, which is the cell lineage. Traditionally, the scientists use the separate 3D tools and record the lineage in a text file, which is cumbersome and error-prone. We created a new tool called LineageD that combines 3D and abstract representations and interactions, along with a machine learning model to support the biologists. In main 3D view, we display the embryo in its current development stage. Second, in the hierarchy view, the cell division history is interactively constructed by the biologists. Every block here represents a single cell at a specific time stage. This stage can be selected by clicking on a given cell or by using a respective slider, and the respective shown 3D structure depends on the level selection in the hierarchy. When the given cell is selected in either of the two linked views, the main view highlighted, and in the target and sister view, we emphasize this cell and its proposed or assigned sister within the context of the rest of the embryo. Traditionally, biologists manually established the lineage top-bottom up. To ease this process, we use the linear classifier to predict likely cell pairs based on past lineage data, one level at a time. Biologists then verify or correct these assignments. Biologists can target a given cell and analyze its neighborhood and correct the assignment in 3D environments. However, for such a big embryo, occlusion issues that affect the navigation exist in both the 3D environments and in the 2D hierarchy tree. To solve this problem, we implemented visibility management techniques. To show the internal structure, we provide means for explosion views as well as cell layer peeling. For the abstract hierarchy view, we made the tree zoomable and provide a word-in miniature thumbnail view to allow the biologist to navigate the tree. Based on their experience, biologists can also reason in a top-down fashion, which we now support in our approach. They then divided the embryo in a few stages based on this shape to establish a part of the lineage tree from the top. This process also introduced constraints for the linear classifier, improving its results. We conducted empirical case study evaluations with 5 biologists and found that during the study, they were largely influenced by their past interaction habits. Also, the tight connection between 2D and 3D representations is essential and necessary for them to make assignments. Moreover, they all benefit from the 2 ways of establishing the lineage, bottom-up and top-down. After the study, all of them said that they appreciated the visual representations of both 3D and 2D and agree that the tool helps them to better understand the embryo's development and are keen on to continue to work with the tool.